So I'm going to give a very brief flavour of the types of coronavirus intervention work that we've been doing at Nottingham. I would say that our interest in coronaviruses was just before the pandemic, mainly because HCV, hepatitis C, which was a virus we worked on before, was pretty much cured, and therefore we had to find other things to do. And so we had a program of metagenomics virus discovery. So we were mainly interested in coronaviruses because of that. But of course, when the COVID outbreak appeared, then we got more involved with SARS-2 research. Okay, so just to remind you, so Dallin would have gone over this, and unfortunately I've stolen his time. But just to remind you all that the progress around treatments has been incredibly rapid. And today we've heard about 2 new antivirals being approved, which have an amazing impact on the rates of hospitalization, serious disease, and death. So molnupiravir, which is, I think, from Merck, which is an direct antiviral. But also, we hear rumors today that Pfizer also have a protease inhibitor that maybe reduces the likelihood of hospitalization by as much as 90%. So that's a huge impact on this outbreak. So I think there are a number of very effective drugs, and clearly very effective drugs are going to come to the fore over the next few months. But you can see here that essentially there are 2 things that you can do. You can either go for an antiviral where you're directly targeting the virus, or you can try and treat the symptoms, and in particular use immune suppression so that you prevent the more serious impacts of COVID-19. So one thing that I'd like to look at is something that is a very safe approach to therapeutics, and that's the use of antibodies, either as convalescent sera, although the data for that hasn't been too awe-inspiring, but particularly the use of monoclonal antibodies. So we know, for example, that Regeneron have a cocktail of monoclonal antibodies that have proved to be very good at reducing the likelihood of infection, but also in serious disease. But one of the problems that we have with monoclonal antibody therapy is the fact that antibodies you can get escape from antibodies. So we know, for example, this was data generated in David Ho's lab over in New York. So if you see here, you've got 3 variants. You've got the original variant, then you've got the alpha variant and the beta. And you can see that the beta variant is pretty adept at escaping the impacts of those antibodies. So that you need a lot more antibody to get the same amount of virus killing. And so this is a potential problem. And this is one of the reasons that Regeneron have gone down the route of using 2 monoclonal antibodies, because you decrease the likelihood of escape. But there's something that I'd like to just very quickly touch upon, and that's some work that we've been doing for a while at Nottingham, mainly in the context of other emerging infections. But that's to look at the potential therapeutic benefit of I did mean to write monoclonal antibodies. That's Richard Banovich's very poor joke. But so why cow monoclonal antibodies? So what we know about the cow antibody repertoire is that occasionally they produce these antibodies that have an incredibly long and complex structured CDR3. So you can see this here in purple in these 7 monoclonal antibodies. So this is a region of the antibody that is cysteine rich, so it's highly ordered, but it also protrudes from the surface of the antibody. And so it can penetrate into recesses, it can get by glycan shields that envelope viruses often use to try and evade from antibodies. And indeed, we've got evidence that these cow antibodies can elicit very broad and potent neutralizing antibody against very variable viruses like HIV. And so we've been carrying out work with cows, immunizing them for a variety of infectious viruses, but including SARS-CoV-2. And so essentially we take the cows, somebody upstairs does some very fancy flow cytometry and delivers us with some single cells. And then we can take those single plasma cells or memory B cells and clone out the antibody variable regions and then express them and test them. And so we've been doing very extensive immunizations with SARS-CoV-2, and we've used various forms of the S protein. So S1, we've gone with full length spike, but also we've gone with trimer as well to try and increase the amount of antibody, but also the breadth. And so what I can show you here, this is a work that I was doing on a different virus, but it shows the principle. And that is that if you continue to boost and inject these animals, eventually you do get a very broad antibody repertoire from these cows. And you can see that in the panel here, that you can see early on in infection, you get a little bit of neutralization against the original SARS isolate. But then when you go further, you can increase that cross-reactivity so that we can get very good neutralization of both SARS-CoV-2, but the original SARS isolate, just by immunizing with SARS-CoV-2. And if you look at the antibody repertoire, what we generally see is an overall lengthening of the CDR3 region of the antibodies. So this is as you mature and you get somatic hypermutation, you increase the CDR3 length of the antibodies that you recover. And you can see that in this diagram here. So here we've got some antibodies that have got exceptionally long CDR3s, as many as 60 amino acids. And then when we check them in a SARS-CoV-2 binding assay, we get good binding with a lot of these antibodies, but we also see potent neutralization. So this is a good route to get very reactive and highly neutralizing antibodies. And at the moment, we're characterizing these antibodies and isolating more. So just to summarize that bit, so hyperimmune plasmid shows certainly broad reactivity. We've got lots of antibodies isolated, many of them with ultra-long CDR3s. They are spike reactive, certainly, and many of them are potently neutralizing. One of the things with the antibodies is that they're not easy to express. So we're doing a lot of work on the expression process optimization. But ultimately, what we're trying to do is to identify a single antibody that can work across lots of different species of the Sarbeca-viruses. In other words, any coronavirus potentially that's using ACE2 or a similar entry pathway, we hope that we can identify antibodies that will be reactive. Okay, so I'm going to move on to some work we've been doing with vaccines very, very quickly. And so we've got some very effective vaccines. They've proven really important. But there are issues with waning immunity. We worry about variants. There's a threat of SARS-3. And of course, logistically, some of these vaccines are problematic. And so we've been working with a Nottingham spin-out company called Scancell, looking at two different vaccines, SCOV1 and SCOV2. So the SCOV1 has got the original strain. SCOV2 is one or more of the variants of concern. And the key features is that this is an identical plasmid backbone, which has been used in humans. So it's already been trialed in humans for cancer vaccines. And importantly, it's a bicistronic plasmid so that we can drop nuclear protein as well as S protein so that we get a good T cell response, but to a conserved region. So essentially where we're at with that is that the vaccines have gone through extensive preclinical work in animals. That data has been very encouraging. So we get good spike neutralizing antibodies, but we also get very good potent T cell responses. And so we've just literally started the phase 1 clinical trial in South Africa. And the first patients were recruited and injected last month. So hopefully we'll have some data about how well they're performing. Okay, and one last thing is to remind you that, yes, we started with metagenomics and virus discovery. We're working mainly on rodents because rodents, we seem to forget them. They're an important source of viruses, but we are also working with some museum samples with the Natural History Museum, looking at very ancient viruses that might be present in those species. So I will leave a list of people who are involved in that, and thank you very much for your attention.